I would like to introduce third speaker, Dr. Kevin McElwee from University of British Columbia. He is Associate Professor of the Department of Dermatology, and he will present an interesting paper. The title is Hair Follicle Mesenchyme cells exhibit immune privilege and can improve islet allograft survival. Okay.
Good afternoon, everyone. So I have a conflict of interest. I now work with Replicell Life Sciences, although I still work at the University of British Columbia as well. But today I'm actually wearing my academic hat. This is an academic study. Hair follicle immune privilege. We've had several great presentations at this meeting about immune privilege and potentially collapse and things like alopecia areata, scarring alopecia, and probably one of the best or simplest elegant demonstrations that hair follicles and parts of follicles have immune privilege. A study done by Colin Yehoda and Amanda Reynolds, where actually they took hair follicles from Colin, dissected them out, dissected the pillar, dissected the sheath, implanted that into the arms of Amanda, and found that there was hair growth. So it's an allogeneic transplantation. So they got hair follicle growth, formation and growth, and also there was very little or no inflammation around the follicle. And they also confirmed that the tissue, the cells that they implanted, were in fact from a male and the epithelium was from a female. So I've been very interested in immune privilege, particularly in relation to alopecia areata. A while ago we looked at a variety of potential factors and how they were expressed in the follicle, in the mesenchyme. We split the mesenchyme up into different regions, the DSC, the cup region, non-bubble dermal sheath and dermal pillar. And we see a wide range of factors which are increased or decreased in expression in appropriately related ways that confer immune privilege. And the one we homed in on was PD-L1. PD-L1 is very potent in terms of regulating and suppressing T-cell activation. We found it in the hair follicle, and it may not be terribly clear, but there is some expression here, the brown stain, right down at the base at the bottom of the cup region, also elsewhere in the follicle of the cup. And when we used the cells from the cup and cultured them and mixed them with PBMCs from a different person, we looked to see what would happen. We saw that there was suppression of the immune response by the PBMCs, and even for that matter, there was actually apoptosis, primarily through promoting caspase 9. So there's actually apoptosis of cells that might normally respond to the cup cells when you do an allogeneic mixed lymphocyte assay. So we wondered whether we could push this further, and just how far we could push this. Can we use the cells potentially for protecting tissue transplants? And we thought we'd go all the way. We'd use a mouse model, which is an autoimmune diabetic mouse model. So you've got autoimmune inflammation to deal with. And then on top of that, we're transplanting islets into the model. So it's an allogeneic transplantation. So you've got two potential responses from the host immune system. Mouse cells in culture, I must admit, I struggle with them a little bit. They're not very good cells to culture and maintain immune privilege, at least not in my experience. Human cells are actually a lot better. But we could find at least some factors that were differentially expressed related to immune privilege. Then there are other factors as well that are relevant for islet transplantation. And so when we did a mixed lymphocyte assay, this time with mouse cells, taking black 6 cells from the cup and culturing those, mixing them with spleen cells from bowel C mice, we saw similar things we saw with human tissues, with human cells, the suppression of the immune system by these cup cells in comparison to ordinary fibroblasts. So we did our transplantation. We took islets, dissected them out from bowel C mice, healthy mice, mixed them with cultured cup cells, and then we put them into mice that were diabetic. We induced diabetes simply by injecting streptozotocin, and this promotes destruction of islets and diabetes onset. And then you track what happens just by looking at the blood glucose levels. And so we saw with our islets and cup cells, there was a prolonged survival rate of the islets, and there was prolonged production of insulin compared to our control fibroblast group, group 2. But eventually, though it has to be said, the islets did not survive. It was not a permanent return to a pre-diabetic level. All of the mice did develop diabetes eventually. And when you look at the location where the islets and cup cells are transplanted to, we actually put them into the kidneys. you can actually see very clear differences in terms of what you see. So by 3 weeks afterwards, you can see the islets and the cup cells very obviously here. You can't really see anything in the control group. If you also look very carefully, you can see there's a lot of blood vessels infiltrating in and around where the implantation site is. This seems to be something that's also helping in terms of getting the islets to transplant. The EGF expression from the cells is also, from the cup cells is also useful. When we looked at the histology and immune histology, we see big differences. Insulin still being produced for 3 weeks in our cup cell transplants mice, nothing at all in our control group. So when we do a mixed lymphocyte assay, I won't actually go through this in detail, but we were able to demonstrate that there's a fairly selective response only to the alloantigens on the islets. So the islets, remember, are coming from BALB-C mice, and so we're taking the splenocytes and we're taking the lymph node cells from the mice getting the transplants and mixing them with splenocytes from other mice as a challenge. So either coming from BALB-C or from CPH, something brand new, never been seen before. And what we see is that a very selective difference. So there is a normal response by the draining lymph nodes from the transplanted mice against the CPH splenocytes. But when we look at the BALB-C splenocytes and how the lymph node cells are responding to those, there's actually a reduction in the response. So it's relatively selective. There's not a blanket immunosuppression. It's actually relatively selective. Why might this be? I can't entirely explain it right now, mainly because we haven't looked into it in great detail. But when we took the cells from the draining lymph nodes and looked what was inside the lymph nodes from the transplanted mice, we saw that there were actually more FOXP3 positive cells. So there seems to be potentially an increase in Treg cells that we're promoting as part of this transplantation process. And so Treg cells are specific, just like effector cells are, for specific antigens. So when we look at the ratio of the Treg cells in the draining lymph nodes from the kidney against the T effector cells, we can actually see a reasonable difference in this. So this might be one way that we're getting to selective immunosuppression and regulation using these cup cells. So the cup cells, they do express immune privilege factors on the surface, also secreting various factors as well. We were able to at least partly suppress alloimmune response and the autoimmune response too, and in doing so we were able to at least prolong allograft islet survival. So I'd just like to thank particularly Dr. Xiaojie Wang, who did most of this work, and the work was done with grant support from the British Columbia Transplant Society. Thank you.